Our next speaker is Jesús Arrieta, commonly known as Chechu. Chechu currently holds a permanent position as a senior scientist at the Spanish Institute of Oceanography, IEO, in Santa Cruz de Tenerife, Canary Islands. He graduated in biology science at the University of Basque Country. After his graduation, he moved to the University of Vienna, Austria, and later to the Royal Netherlands Institution for Sea Research, the Netherlands, where he obtained his PhD degree at the University of Groningen. After a postdoctoral period, he held the Ramón Casillas research position at the Mediterranean Institute of Advanced Study, IGME-CSIC. Between 2015 and 2017, he worked at the Red Sea Research Center of the King Abdullah University of Science and Technology, Caus, Saudi Arabia. And his scientific interests focus in marine microbial ecology and the role of the microbe in the functioning of the biosphere and the regulation of climate processes. So let me...
Okay. Well, first of all, I have to say that actually this presentation should have been given by my colleague Isabel Ferreira, who has been involved in the Vulcana project since the very beginning. I entered this very much later, so most of the data that we are going to present are her data. Yeah, so close. Okay, so I'm going to talk about mainly microbes related to volcanoes. And talking about microbes related to volcanoes, well, first of all, I have to say I'm not a geologist, so probably I'm going to oversimplify and abuse some concepts, so sorry about that. Okay, so, well, talking about microbiology is linked to water, of course, as all life. So my talk is going to be mainly about water that goes in contact with a volcano, right? Hydrothermal fluids, these kind of things. Well, the whole trip here, whole trip of water starts when cold seawater sinks into the crust, into a crack, and then, well, very rapidly, as it goes into the crust, it loses oxygen and potassium very quickly. Then calcium sulfate and magnesium are removed, and then, well, as it warms up and gets to really warm places, it gains some sodium, calcium, potassium, and later some metals. So we end up with something completely different, the water that has lost some minerals, has gained some others, which is overheated and at some point comes back to the surface. Okay, there is something that was covered at the top. Well, anyway, it can leave the crust either as a black smoker, let's say, with carrying all these metals, or it can just mix with seawater and leave somewhere else as a white smoker or maybe as a diffuse flow. Right? This is what we have: a modified water carrying lots of reduced products like sulfate and these kind of things. And then, well, the idea is that well, we can find different types of bacteria associated to these places, and we can find these bacteria when the things are as hot as 120 degrees. And depending on where we are, depending on when we are, well, we find different types of bacteria, like the ones we can find, for example, in black smokers. We have lots of sulfur oxidizers. We have sulfur oxidizers all over the place. We have also some oxidation of metals that can be abiotic, just produced by the contact with oxygen and cold water, but also mediated by microbes. And then we have also the formation of microbial mats and things like this that are going to be growing on these reduced products released by the hydrothermal activity of the volcano. So basically, to simplify this very schematically, we can find this kind of processes from 2 degrees, which is would be the temperature of the ocean, of the deep ocean, down to 121 degrees centigrade, which is so far the upper limit for of temperature for life that we have been observing. So depending on that, well, at the very hot end of the processes, we have things like reducing conditions and lack of oxygen, so anoxic processes, and we have the reduction of CO2 by hydrogen, for example, mediated by different bacteria and archaea. And then as we progress to aerobic conditions, we have other processes like oxidation of as oxygen becomes available, we have other processes like oxidation of sulfates, for example. Right? This is a bit the scheme and the different organisms that you will have involved in these processes. So the importance of these processes is that basically, in the absence of light, even you can generate a trophic food web. Right? So you start with something with chemoautotrophs that are actually growing on things that are nasty and toxic for the rest of the organisms, right? Like sulfite. And we end up producing organic material that can be consumed by protists, by zooplankton, going to larger organisms. It can eventually end up in fish even, right? But we also have interactions with viruses and with heterotrophic bacteria that are going to consume part of the organic material that is generated there. And well, this gets more and more complex. At some point, it gets even weird. Like in this example that we have here, the interaction of viruses with chemoautotrophs, for example. It has been shown recently that actually viral lysogeny, which is different from lysis, and I will explain this now. It's prevalent at these places where you have diffuse flows of sulfur. And actually, well, you probably know the lithic cycle. So a virus enters a bacterial cell and kills it immediately, takes over the machinery, produces new viruses, and kills it. But then you have lysogeny, which is a bit more complicated process. The virus injects its genome in the cell, and very often it carries also parts of the genome from the original host that it just killed. And sometimes these parts of the genome come with something useful, like for example the ability to oxidize sulfide. Right? So as long as they are happy together, this DNA will integrate in the DNA of the host. And as long as the host is happy and just reproducing itself together with the virus that it carries, well, this is just giving the host a new ability that is useful for it in this environment. And if conditions go, if sulfide is not available or conditions go difficult for reproduction of the host, it can eventually kill it, release it, and go find another host. So this process seems to be prevalent in this kind of environments, and this is something relatively new. So now going back to coming closer to our favorite volcano so far, El Hierro. Well, we have heard about the submarine eruption in October 2011. In the beginning, we had this plume that is very similar to what you can find close to a black smoker, just lots of metals and sulfide and other things, lack of oxygen, these kind of processes. And later on, after the eruptive phase, we are now in the degassing phase, where we can still find hydrothermal fluids, inorganic nutrients being pushed by these hydrothermal fluids, sulfide, iron, and other metals, right? Okay, so before I show you some data, I have to show you how we do it, just to give you an idea where the data come from, and maybe to give you an idea what they mean. So this is basically what we find under the microscope. We can find bacteria, these big dots, and viruses, which are the small dots. So we have many more viruses than bacteria. This happens everywhere. But the salient feature here is that, well, these bacteria probably, belonging to hundreds, maybe thousands of different species, and we don't see any difference there. So the microscope doesn't help. It won't tell us what they are doing or who they are. Right? So we have to use something else, and this is so far the most used approach, which basically consists of you take your sample from seawater, or from the microbial mat, whenever you want to study the microbes, even from somebody's skin. The methods are the same. You extract the DNA and then amplify selectively one fragment of a gene that is common to everyone, like the 16S ribosomal RNA gene is the most popular. So you can amplify this gene for every piece of DNA that you have extracted from the community. Then you sequence this, and then you compare it to a database where you have this gene for all the known bacterial species, right? So by this comparison, you can identify where this DNA is coming from, and you can come up with a relative abundance of these. We are not going to call them species, we are going to call them OTUs, like operational taxonomic units, because sometimes it's very hard to get to species level, right? And to keep the taxonomists happy, we have to just use this kind of euphemism, OTUs, right? But this is basically the idea. So when you do this, you come back to the samples that we took in El Hierro, before, during the eruption and after the eruption, we find things like this. If we just do a PCA analysis very quickly of just taking all the possible different species and their relative abundances, and we plot this, well, we find that in the epipelagic, so in the first 200 meters of the water column, we find communities that are very, very different from those that we find after the eruption, right? So this is the first thing that we see. When we look at these communities, actually there is not so much difference in the number of different things. So most of the species of the OTUs that we find there will be common, right? But we have a few that appear only during the eruption. For archaea, you know what archaea are? It's just something similar to bacteria, but they are radically different, right? Evolutionary. But they are as small and looking the same as archaea. In the picture that I showed you before, you won't be able to tell one bacterium from one archaeum, right? Okay, so what do we find there among the minority, this small minority of things that are different? We find like epsilon proteobacteria that are often associated to sulfur oxidation in hydrothermal vents. And here you have these things that are in black, these OTUs, right, that were isolated from our seawater samples. And then around them, you see another known clones from different places, and you see they all come from volcanic eruption, hydrothermal vent, these kind of things. So they are the usual suspects that you are going to find around a volcanic eruption. We found some members of the candidate division, this ZB3, which is now known as class Candidatus marina margulis bacteria, which we know that they are associated to sulfur oxidation, but there is no single representative in culture, so that's why it's not an accepted species, but a candidate species. So it's supposed to exist, but we don't have it in pure culture. And then we also found some members of the Zeta proteobacteria, which are often associated to the oxidation of iron. Right? All these things in the plume that we won't find later on in the seawater. Okay. Then, after the eruption, another group came to Tagoro, to El Hierro, to the submarine volcano, and found this kind of carpet, this thing covering, this white thing covering the whole surface of the ocean, and this thing around the volcano. And this thing, well, is actually, you know, when you look at, you know, from the distance, and when you get closer, it looks like hair. The authors of this paper, they called it Venus's hair. They had like poetic day. So, and what they did is, well, instead of taking the way that I showed you before, it took the more the brute force way. So they extracted the DNA, same as before, but instead of going for one single gene, they sequenced everything. Yeah. All possible genes of every single organism present in the sample, right? And then, well, they came up with this whole sequence of everything. But since this white stuff, this white fluffy stuff was dominant everywhere, they got lots of reads, lots of these fragments of genes that belong to the same organism. So they assembled them and they came up with this genome of a theoretical organism that they don't know it exists or not, but it looks like something that was not known to science before. And if you look to closest relatives using different genes, it shows up here, you know, very separated from everything that is known, you know, from this kind of environment. So they came up with this thing like it must be a different species. Right? But they not only found that, when they sequenced everything, they found also genes from other organisms, like from larger organisms. I told you before, this kind of environments are not only giving food to this bacteria, but they are also giving food through the bacteria to larger organisms. You know, they are keeping a whole food web behind them. Okay, since this candidate organism, which is called Venus's hair or Thyolava veneris, is not available in pure culture, we just went there and we collected some samples and we gave them, we put them in these petri dishes, and we gave them to eat only sulfite. The only thing they have there to eat is sulfite and maybe the agar. So some bacteria ended eating up the agar, and we discarded those, but these guys here are growing only on sulfite. So that's why, since this is the only thing they have to grow, and they grow, we conclude that they are doing this process. No, we are just in the process of identifying them, but we already have them in culture. We don't know whether this is this hypothetical organism or somebody else, which is also in this process, but this is it. Okay. Now for something different, going to last year's eruption in La Palma. Well, we have heard about the formation of this lavic delta. We were there just a bit after the eruption started, so we couldn't take samples before the ashes went in the water. So we are going to have a very hard time, you know, seeing what the actual effect of the ashes is, because the ashes are everywhere. You know, cover a very large area around the island, and of course we cannot find the oil. It won't be easy to find a good control for this, right? A place that is similar enough to the place that we are studying and that has been not exposed to ashes, right? But we can just try to do this study in the water, you know, independent of the ashes, to see whether we find something else, something, you know, some other effect from the materials that are being released by the lava entering the ocean. And this is basically what we got. We just sampled in all these places and the expected place for the lava to reach the ocean. And of course, this is sometimes an exercise of you know, guessing. Although we had some clues from our colleagues. Well, the thing is, well, we took samples all over the place before the lava hit the water, and then we tried to get as close as we were allowed to take samples at the same place. Sometimes this was not possible because the lava was covering the place where we took the initial sample, and sometimes it was just not safe to go as close. So the sampling is also a bit tricky, but nevertheless we got some results. Well, most of the samples before and after the eruption will show up here, you know, like they are not very different. We didn't manage to get close enough or to get, you know, to the place where we had this thing. But some of the samples after the eruption were clearly different. And also when we used a drone approach the lava delta and take some surface samples, we also got some very, very distinct communities, right? If we look at the distribution of different organisms, you see, well, we are sort of lucky because before and after the eruption, most of the samples look the same, right? So there was not a large change due to just to time, because also these communities change over time, right? As the season progresses. So this is a good thing. And then we were lucky to hit some places where we had like more archaea than we expected. For example, this is a good indication of there is something related to the volcano going on there. Or these are the drone samples, just an average of them, which contain a lot of gamma proteobacteria, this dark pink stuff, which can also harbor some of these sulfur oxidation genes. But this is something that is work in progress right now. If we go to, just to show you another example, if we go to these places in El Hierro, for example, at the bottom, in this carpet of organisms that are covering the place, and we get to one of the obvious places where there is a lot of hydrothermal fluid coming out of the floor. Well, you find these thick mats, these microbial mats, where it's very likely that is happening this process of sulfur oxidation, right? And this is just a comparison with the samples that I showed you before. So here you have the La Palma samples from the surface, some surface samples also from El Hierro. These are the different ones, the ones that are from La Palma that were different from the rest, the drone samples, and then this bunch of samples that we took using the ROV just straight from the mat. And here we find again these brown bars, a prevalent community of Zeta proteobacteria. Remember the ones that I showed you before that were associated to oxidation of metals? You find them only in these mats, you don't find them anywhere else, not in La Palma, not in El Hierro. And this is another view of the same thing. Well, these are ROV samples where you find the Zeta proteobacteria, this big bar here. They don't show anywhere else, right? This is a bit an overview of what I have. As I told you, this is work in progress. We have more questions than we have answers right now. But, well, if you can come up with another intelligent question that we can use, you're welcome.
Thank you so much, Jesús Arrieta, for your talk. It has been really, really interesting. And also to Isabel Ferreira, that is online. She couldn't be here, but all is her work and also the work of Jesús Arrieta is really, really interesting for us. Any question in the... Okay.
I have a quick comment and a question, if that's okay. During the Hunga Tonga eruption, we actually saw the identical white microbial mat as the first successor. So I thought that was quite interesting.
Which one?
The Venus hair white microbial mat, we saw identical in the Hunga Tonga eruption as the first successor of that deposit.
Yeah, well, this some of these sulfur oxidizers, they have the same aspect. They make these long threads. They also have like a sheath, and these filaments inside. And they are also, if you have tried to collect them, they are like incredibly, fluffy and very, very delicate. If you approach them and you move the ROV just this quick, they will fly away. They are stuck to the surface, and I don't know how they keep themselves there. But the moment you try to catch them, they are gone. But it could be Venus's hair, could be something else. Of course, this organism, even if it's new to science, must have come from somewhere else, And if you see how rapidly they develop in this newly, after an eruption, the capacity for dispersal, it must be enormous. They must be able to survive, for very long stretches, transported by the ocean in places where they don't have no food at all.
Right, and then a quick question, if that is okay. During an eruption, as some of these vent communities, they might become unstable. do you see a breakdown in that relationship, in the symbiotic relationship of the chemosynthetic organisms?
Well, you mean in the water column?
In the vent communities, do you see a breakdown of that symbiotic relationship?
Well, these mats, they are organized usually, you know, within in depth. So you will have like a very organized community there. Is this what you mean? So you will have different organisms at different depths, depending on the amount of oxygen that it reaches. You know,
kind of like I meant more like in coral and zoo and sand fillet, how they expel them during when the pressures become too much, when the pH becomes too much, or the temperature. Do you see a similar reaction in the vent communities during an eruption?
No, well, it's a bit what I showed you before. There are these ranges, you know, like for example, if you have these sulfur oxidizers, they need oxygen. So if you know, if the eruption is really going strong and there is no oxygen, you know, the pH is very low and you know, and it's very hot. The problem is there is no oxygen, so they cannot do what they do. Of course, they all have their limits also in terms of temperature. So it's, yeah. This is the graph I showed you before, with the limits of different processes depending on temperature and oxygen concentration. Mm-hmm. So this is what you expect to find depending on the conditions.
Thank you. Just a quick question. Is there any specific pattern for this development of the Venus hair? I mean, you said that it has to do with the sulfur, but except the sulfur, I mean, the distance from the vent, the condition of the water, the depth, the type of the rock, the whatever, the pH, the acidity, the alkalinity. I mean, or is just sulfur?
This haven't been explored, you know, like the limits of this organism, in part because you cannot. In part because there is not much of this organism that we can use because it's not in culture. We cannot do the experiment, right? But yeah, where we find it is where we find these mats is associated to cracks, to places where you can see that the fluids are coming. You know, like you see this blurred thing in the images, where the hydrothermal fluids are coming really strong. This is where you find the mats. So it's, yeah. We know that it's using sulfate and probably other things, but the main nutrient for this organism must be sulfate. Yeah.
Another quick question for Jesús Arrieta. Yeah.
I didn't expect so many.
Can you turn next to the camera?
Okay, yeah.
This is just something I'm curious about because I don't know much about biology. So, where do these organisms come from? Like they develop in these extreme environments, but firstly they have to come from somewhere.
Well, I suspect that they don't come from very far away, actually, because, you know, in the Canary Islands, we know a few places where there is this hydrothermal activity and where there are mats, and there are probably many more. But it's just that we don't know them. But if you see that wherever you find this kind of emissions, you find the mats, probably they don't come from very far away, but their potential for spreading is very high.
Thank you.
Thanks. First, I want to say congrats because it was like a very inspiring presentation. And my question is, I know this bacteria, it's like new, you just discovered them, but do you think it has like a potential to be used as a bioremediation for example, leaks of sulfur in the ocean, for example?
Well, like sulfate is not very stable in the ocean, right?
Yeah.
If it's not oxidized by bacteria, it will be oxidized abiotically. So, yeah, I don't know if you could imagine of a scenario where you can contain, let's say, the emission to a rate that can allow for this to be used as an agent for bioremediation. Yeah.
Thank you. So we have to move on. So the next, thank you, Checho, again.